Welcome to ISL Enabled Virtual Lab of CSIR.
How is paralysis diagnosed?
By movement, by deficiencies in movement sense due to sense. So you have to stimulate, right? Stimulate an individual or a patient, and the normal response should be movement, and that does not happen. Now again, movement, we often think of movement as walking, but for even just for me to blink, it's movement because muscles. For my food to get digested, there is movement because there's peristalsis in your intestine. Need not be movement of walking. So diagnosis would be, there should be, under a normal case, normal situation, a controlled person should be able to move that particular part of the body or organ, while this individual or animal model is unable to do so.
The next question I have is, what are the mental effects of paralysis?
The paralysis has occurred because certain brain regions have become defective, whatever, lack of nutrition, lack of blood supply, injury, degeneration due to various reasons, infection, right? All of these reasons can lead to a breakdown of cells or ill health of cells in certain regions in the brain, which are important for motor function. Now, we think that these regions are very important for motor function. It doesn't mean that these don't have any role to play with our emotional or cognitive abilities. We can't rule that out, correct?
Is there any way to prevent paralysis?
Yeah, again, like I said, paralysis is the end result, like fever. Is there a way to prevent fever? I don't think there's one answer to that, because paralysis can occur for various reasons. And if we are able to somehow keep an individual healthy or an animal healthy, then we reduce the chances of paralysis. That's all.
From the discussion so far, we've realized how complex paralysis is. So we were wondering how you actually study this phenomenon in your lab. We know that you work with fruit flies, right? So how do you do the research?
Okay. So I must again clarify here. It turns out that we studied a condition which also is associated with paralysis. Okay. One of those early collaborations was the work that Anushka did. Now it turns out that in humans, as I told you, as you guys were discussing also, that there is a gradual progression where motor functions are gone in people who suffer from that condition. We needed to model that in the fly. We chose what is known as the neuro-muscle junction. So not neuron-neuron connection, but neuron-muscle connection. In the fly neuromuscular junction, this synaptic region becomes big and expanded. Here is a place where a neuron is meeting a muscle. Neurons and muscles are needed for movement. So we chose a motor neuron meeting a muscle wala circuit to do our studies. How do you do that? You can do it. So another thing to remind you is we work with Drosophila melanogaster, the fruit fly, which you can commonly see even in your homes. If you have a fruit that's very ripe, you'll see them buzzing around. So what Anushka did in our studies is we focused on the larva most of the time. So the larva, have you seen? No, you probably haven't. Actually, next time you see a rotting fruit, take a closer look at it. If flies have laid eggs on it, in a few days, you will see these little almost translucent, creamish white little wriggly worms inside these rotting fruits. So we grow them, of course, in the lab, and in a glass dish, you can fillet them out. You have these larvae in these dishes, and with a very thin dissection equipment, you can cut them out in such a way that you can fillet them out like this, and their gut and everything is gone, and only their body wall is left. So imagine a tube like this, okay? And then you just cut it out. So now all you got to do is do this, and you have like a flat fillet, and the muscles are, flattened out. And it's, as I said, very, very stereotypic organization. So larva after larva after larva, you can dissect out, and they have the exact same, muscle band number 1, 2, 3, 4, and motor neuron A, B, C, D, and how it's supposed to innervate. So you can go exactly to the same region in sample after sample after sample, and you have a very stereotypic preparation. So under the microscope, you are seeing this neuron to muscle junction.